Hello and welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here's what I bring to you all from the world of medicine. A study that reveals how the ovarian reserve is established. Researchers define the epigenetic machinery that governs the establishment and function of the mammalian ovarian reserve, providing molecular insights into female reproductive health and lifespan in a new study published in Nature Communications. In human females over the age of 35, you see a decline in fertility, said lead researcher of the paper. Our study may give us the foundation to understand how female fertility is established and maintained at the molecular level and why it declines with age. When the ovarian reserve is established, all the oocytes in primordial follicles pause their development and can remain in such an arrested state for decades. The main question is, how can these cells be maintained for decades, said the researchers. Using mouse mutants, the team found that the pausing of this oocyte transition phase was mediated by a group of proteins called the polycomb repressive complex 1 or PRC1. PRC1 suppresses the development process called meiosis that occurs prior to establishing the ovarian reserve, thereby ensuring a proper gene expression program in the ovarian reserve. When the team created mouse mutants with depleted PRC1 machinery, they found that the ovarian reserve could not be established and the cells underwent cell death. These results strongly implicate PRC1 in the critical process of maintaining the epigenome of primordial follicles throughout the protracted arrest that can last up to 50 years in humans, they noted. According to Namikawa and his colleagues, deficiency in PRC1 functionality may help explain cases of premature ovarian failure and infertility in humans. Higher risk of dementia if an upper heart chamber is abnormal. Structural or functional abnormalities within the heart's left atrium with or without symptoms may increase a person's risk of developing dementia later in life by 35%, according to new research published in the Journal of the American Heart Association. Dementia risk increased even among those who did not experience atrial fibrillation or stroke, two conditions known to be associated with dementia. Atrial cardiopathy is associated with an increased risk of stroke and atrial fibrillation, which are both linked to an enhanced risk of dementia. In a study led by Michelle C. Johansen, research team aimed to determine the relationship between atrial cardiopathy and dementia, and if so, whether it's independent of atrial fibrillation and stroke. This analysis uses data and assessments gathered during participants' fifth ARIC clinical visit between 2011 and 2013 as a baseline and follows the participants through their sixth visit between 2016 and 2017 and their seventh visit between 2018 and 2019. The current analysis included 5,078 of the 5,952 participants who returned for their fifth clinical visit. 59% were female and 41% were male. The group of 5,078 were an average age of 75 years old and 21% self-identified as Black adults. During their 5th, 6th and 7th clinical visit, the ARIC participants were evaluated for cognitive decline, indicating dementia. The analysis of the collective health data found that throughout the more than 70 years of follow-up, 763 people developed dementia and 1,709 had atrial cardiopathy. The participants with atrial cardiopathy appeared to be 35% more likely to develop dementia. When the researchers adjusted for participants who experienced atrial fibrillation and stroke, even after accounting for other vascular risks, they still observed a respective 31% and 28% increase in dementia risk in patients with atrial cardiopathy. The researchers suggested that a state of atrial cardiopathy leading to dementia is not a result of atrial fibrillation or stroke alone. A new drug candidate which fights off more than 300 drug-resistant bacteria. Gram-negative bacteria are especially difficult to treat because they have strong defense systems, tough cell walls that keep most antibiotics out, and pumps that efficiently remove those antibiotics that get inside. The microbes can also mutate to evade multiple drugs. Furthermore, treatments that do work aren't very specific, eradicating many kinds of bacteria, including those that are beneficial. So Paul Hergenrother and colleagues wanted to design a drug that could infiltrate the defenses of gram-negative bacteria and treat infections while leaving other helpful microbes intact. The team started with an antibiotic that was active against gram-positive bacteria and made a series of structural modifications that they believed would allow it to act against gram-negative strains. One of the modified compounds, dubbed fabimycin, proved potent against more than 300 drug-resistant clinical isolates while remaining relatively inactive towards certain gram-positive pathogens and some typically harmless bacteria that live in or on the human body. In addition, the new molecule reduced the amount of drug-resistant bacteria in mice with pneumonia or urinary tract infections to pre-infection levels or below, performing as well as or better than existing antibiotics at similar doses. The researchers say the results show that fabimycin could one day be an effective treatment for stubborn infections. The surprising way in which multiple sclerosis drug works. Cells that make myelin sheath are sensitive to adenosine triphosphate or ATP and nitric oxide or NO, molecules that are present in high amounts in the blood and brain lesions of MS patients. Red blood cells can release NO directly, but they can also simulate NO production in the lining of blood vessels by releasing ATP. NO can then go on to damage nerves in MS patients. Zinc C-peptide, which is secreted by the pancreas along with insulin and albumin, are key players in the latter processes, and they can latch onto red blood cells. Because interferon beta can bind zinc, it seemed possible that the drug helped patients by sopping up this mineral, so Dana Spence and colleagues wanted to investigate further. In lab tests, the researchers found that red blood cells from MS patients bound more zinc, C-peptide and albumin than cells from control subjects. Treatment with interferon beta reduced this interaction in MS samples down to control levels. Albumin boosted zinc and C-peptide binding to MS red blood cells, and this effect went away with interferon beta treatment. From this data, the researchers conclude that it's likely that the drug is inhibiting albumin binding, keeping it from delivering its cargo of C-peptide and zinc to red blood cells so that NO can be made. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.